Afternoon. It's a pleasure to be here today to speak to you about Arbor Biotechnologies. I joined Arbor about a year ago from Sigalon Therapeutics, where I was COO of that company. Prior to that, was at Pfizer for a better part of a decade, very involved in cell and gene therapy. Was COO of their regenerative medicine group in the UK. And before that, I'd done strategy consulting and was a Ph.D. scientist at Harvard way back when. So Arbor is a next generation gene editing company, and we are advancing genetic medicines based on our wholly owned CRISPR editing systems. And these have been derived from our machine learning AI-driven platform, from really scouring the metagenomic data to identify interesting differentiated CRISPR systems. It's allowed us to build the most extensive toolbox of proprietary editors in the industry. And this gives us the ability to really focus on the disease pathology and then bring the right editing approach to bear against that pathology. We've got a highly experienced team that know how to deliver and bring these new novel therapies into the clinic. We've got a series of these tools that allowed us to build an internal pipeline focused on liver and CNS. Our first IND target for the end of next year. And we've raised over $300 million to date. The team here is a critical part of our success. First, our scientific founders, Feng Zheng from the Broad Institute, one of the CRISPR pioneers, and David Walt, founded the company based on the premise that Cas9 wasn't the best nuclease found, it was the one that worked. And what if we were to go scour the metagenome and identify novel differentiated systems and multiple systems to allow us to really attack diseases based on the genetic pathology. With that, we've been able to build this really important discovery platform. David Ting, our Chief Technology Officer, leads these platform discovery efforts. John Murphy, our CSO, and Pam Seikoitz, our COO, come with extensive experience in gene editing and gene therapy from their experience at Pfizer and Bayer, and Editas. And then between the three of us, so John, Pam, and myself, we have over 20 INDs in this novel advanced therapy space. We are well positioned due to a very large $215 million Series B round we raised last November, co-led by Temasek, TCG Crossover Group, and Alley Bridge Group. In addition, Arch led our Series A as an important investor in our B, and Vertex is also an important investor in the company. On the partnership side, we've accomplished multiple partnerships, two of which have been announced this year, those being with TCR Squared and Edigy. And then we had a partnership with Lonza last year and Vertex last year, and then a previous Vertex partnership a couple years before. So our approach is to really have this breadth of editing tools that allows us to really accomplish whatever we need to in the DNA. For example, the nuclease is the chassis. So with that nuclease chassis, we're able to build on that to do different editing approaches, whether it's SNP corrections, corrections of variants, or even whole gene insertions, using a variety of coupling of this nuclease chassis to a reverse transcriptase, for example, or to a whole gene cargo in the case of a CRISPR transposase. We really position ourselves as a next-gen editing company where we feel we are well positioned to lead in this space with this bespoke wholly owned Arbor nucleases and various precise editing approaches we have, in addition with the ability to write and replace whole genes in the future. From a competitive perspective, we can certainly play in many areas. You can see that depicted here. And, you know, we do, our IP and our work is very far from the Cas9 space, which allows us to really identify and differentiate ourselves. One important distinction is you'll notice in the ex vivo space, we've had multiple partnerships. That's on purpose. We feel very strongly that a company to succeed should focus on a single platform. In this case, we're focusing on our in vivo genetic medicine platform and not going to do ex vivo cell therapies on our own. We will partner in that space. Our discovery engine really is built on a very large database. In this case, over 3 billion proteins have been indexed from both proprietary and public sources of data. We then comb through these systems, identify systems of interest, and bring those to bear into our high throughput bacterial system where we characterize each of these to identify the size. What is the PAM or the binding motif? What does the off-target profile look like on the system, et cetera? By characterizing these systems in a bacterial high throughput approach, we can then pick and choose those systems that are most differentiated to bring into our mammalian editing platform. In this platform, we then further characterize the systems and engineer them to bring them up to high levels of editing activity. With that, we've got 7 proprietary editors to date, and that number continues to grow. That's allowed us to unlock a suite of editing capabilities you see here on the right-hand side, from RNA targeting nucleases all the way through to CRISPR transposases. Now, this is a phylogenetic tree of all the families that have been discovered in the literature in orange, and then in blue are the families that Arbor's discovered. And you'll notice there are many, many nuclease families we've discovered that are completely novel clades. Over 60 nuclease families we've identified to date. This has allowed us to build a really strong and large and growing IP portfolio around this work. In addition, not only is discovery important, but the ability to engineer. And so in the example shown here, we've been able to generate almost a 40x improvement in editing efficiency from wild-type nuclease to achieve therapeutically relevant levels of editing efficiency. And that's an important piece of the puzzle, because then we can not only discover, but we can also engineer and create novel nucleases that can be efficient at editing in mammalian systems. Now, having a toolbox is important, and that's because we can select for different cut types. We can select for high specificity systems. We purposely have looked at non-commensal sources to get away from any pre-existing immunity. We've also looked at AAV deliverable systems, and then finally had a broad PAM approach, which allows us to really target 100% of the DNA as opposed to pieces of the DNA if you have one individual nuclease. This toolbox approach allows us to really work backwards from that disease pathology. In this case, it was CNS disease. We can see the characteristics of the disease here, and then we can then bring to bear various effector elements that are tailorable from our portfolio, whether it's the PAM required, the guides, the effector needed, et cetera, and pull the appropriate pieces from our toolbox to make sure we have a bespoke therapeutic for that disease pathology. As I mentioned earlier, our focus is on the genetic medicine side, where the nuclease is the therapy. We can tailor that nuclease to the disease and build our own portfolio of programs. We also have a partnership with Vertex in this space. And as I mentioned previously, engineered cell therapy is an area of partnership for us. There's many players that want access to a nuclease, and nuclease is a key building block for the therapy, but we are not going to develop our own ex vivo cell therapies at this time, and we'll partner extensively in this space. The way we've thought about our portfolio in the in vivo medicine space is really identifying areas where there is clear delivery available. So liver is a good place where we know we can deliver via lipid nanoparticles today. In addition, diseases where there's clear clinical and regulatory approaches. And from our perspective, diseases where we can show differentiation from the antisense or RNA-based knockdown approaches or first-generation knockdown approaches. You can see we've listed 2 diseases here. And our approach here is to do what we call a knockdown plus. We'll combine a knockdown with some other editing modality, whether it's a knock-in or some other approach that allows us to show the differentiation as well as the utility of these next generation nucleases. From there, we can expand the platform, moving to liver, where we can bring in more precise editing approaches, as well as bring that knockdown plus strategy into the CNS, where now the risk shifts to delivery. And by carefully selecting our lead indications, we can utilize existing AAV approaches for delivery, while that delivery to broader areas of the brain continues to evolve quickly. Our pipeline is emerging. As I mentioned, our first IND is targeted for end of next year. You can see we've got several liver programs, a couple CNS programs, as well as these undisclosed programs we have with Vertex. Now, from an upcoming key milestones perspective, it's important that we continue to drive towards our first in clinic for end of next year. In addition, we need to bring the rest of our portfolio forward, particularly our CNS programs. This year, we will see data in NHPs, which is an important translatable milestone for gene editing. In addition, we have regulatory meetings concluded and also slated for later this year. And then finally, we are going to continue to invest in our platform. We want to continue to make sure we expand our library of novel modification tools. And this is an important part of our go-forward strategy in the future, to continue to expand our toolbox so we can remain at that bleeding edge of this technology, which is rapidly evolving. So in conclusion, Arbor is a next generation editing company. We are focused on delivering and developing curative next generation genetic medicines. We are developing our own portfolio of internal, the wholly owned in vivo genetic medicines, and we're partnering with leading companies for the best ex vivo cellular therapies. Thank you very much. Goodbye.